All right. Good day to all of you, listeners. I'm presenting on hyper-variable segment 2 of mitochondrial DNA and its relationship to two ethnic groups in Nigeria. That's the Asholio and the Atekai ethnic group in Nigeria. Um, the One of the most noticeable question that we always ask ourselves is the history of the humans and how these humans were able to migrate from one point to another. And in fact, there are so many experts that made their contribution on the migration of humans, being able to show one or two evidence. And the most important is the anthropological aspects of understanding the human and being able to know how civilization come about. Now, one of the most important technologies that has been used to understand the origin of the human is in the field of genetics, understanding the human genetics by studying the DNA of the humans. And the most important marker that has been used in this study is the haplo characteristics of the genes to study that, which gives us an idea of the phylogenetics and the phylogeography of these humans. Now, part of the statement of this work is to give us an understanding, okay, of the location and the ethnic diversity of the Asholio and the Eteka ethnic groups. These are two ethnic groups in Nigeria. They are basically located in the north part of Nigeria. And this population, they look on the outside to be synonymous, homogeneous, because they share the same language, same culture, and even the same geography. But these two ethnic groups tend to see themselves as distinct from each other. So that is one of the thoughts that inspired me to carry out this research work. Now, we're able to carry out this study by applying the mitochondrial DNA, using the mitochondrial DNA to test the relationship between these two ethnic groups to see if there are any gene flow. Is there any similarity between these two ethnic groups? So we're able to do that. So one of the justification of this study is because a few of this study has been done in Nigeria. So hence the need to carry out this study to really ascertain the similarity between these two people. Okay, so this will provide enough needed information to the existing data already on studies done on the human genome in order to ascertain similarities between ethnic groups in Nigeria. So we hypothesize that there will be a shared genetic structure between these two ethnic groups. The aim of this study is to corroborate the origins of these two ethnic groups and to be able to see if there are any shared similarities among them in these two ethnic groups. So the objectives of the study are: we are going to we are isolated mitochondrial DNA, and then we study the hypervariable 1 and 2, even though in this study we are considering the 2. And then we are able to identify and compare any polymorphism. And then we compare it with the device reference sequence. And then we assign each of the individuals to a haplogroup. We characterize the haplogroup, and then we're able to investigate the genetic variability of the population. We compare the sequences with other sequences that have been published. So Buka swab were used to collect the uh, uh, DNA from the population, uh, where the individuals were asked to wash their mouth, and at least they have not eaten for about 20 minutes. So it was done early. The study was conducted early in the morning, where the Buka swab were collected. And then these Buka swab were stored in ice for proper transportation to the lab where the extraction is done. Now, DNA extraction was done using the AquaPrep DNA extraction kit, where the DNA was extracted and then purified. After the purification of the DNA, we are ascertain the purity of the DNA using the photospectrometer, where the DNA was ascertained. And then it was then prepared for sequencing, where we polymerize the hypervariable 1 and 2 segments using the forward and the reverse uh, reagents in order to polymerize the area of interest, hypervariable 1 and 2 segments. These are some of the equipments that were used. So after the polymerization, we gel electrophorose in order to see the various segments. So about 20 males were used, males that are unrelated. These subjects were taken from the village where these two ethnic groups were located. So we took it, having taken consent from the population, as well as ensuring that we are certain that these individuals involved in the study, we are members of this ethnic group. Okay, so both verbal uh, consent and assertion we are noted, okay, to ensure that these males are either the Sholio or they are actually the attacker ethnic group, okay, from the last 2 generations. So this is a sample of what was done. Sample the extraction, then we polymerize, and then we sequence the DNA. The DNA. After that, we're able to find the differences in the sequences that we are determined by comparing the nucleotide position to the reverse Cambridge data. All intra-population diversity analysis were done using the DNA speed, the allopin, the XSTAT were all employed. And then the FST distance to check the relationship between these two were done. And then it was considered to be between 0 to 1. So all of this phylogenetic tree was also formed to see where each of these ethnic groups fall, using the Tajima's D and the full neutrality test. All were all employed. Analysis of molecular variants was also employed in order to see the genetic variation within these two ethnic groups. Principal component analysis was done to identify the patterns in the data generated to express the data in a more clear way. So these were the results gotten. This is the table up here shows the result of the DNA collected. Okay. for both the hypervariable 1 and 2 segments. So we're able to see the level of purity. Okay, so from the data gotten, okay, we show that the DNA gotten was highly pure at the level of 1.85 plus or minus 0.17. So it shows the opacity density of the DNA obtained, and it was very significant. A high yield of DNA was obtained. And then this is the uh, result of the gel electrophoresis. You know, we're able to obtain our DNA, okay, for the hyper available 1 segment of 320 base pairs, and then for the 2 of 260 base pairs. They were noted in the DNA collected after the gel electrophoresis. Now, this is the result of the sequences. Okay, you can see the spike, okay, of the sequences for both the hypervariable 1 and 2 segments. At least we're able, since the DNA obtained was very pure, we're able to see less of noise, okay, less of noise, uh, so we're able to read our sequences as they spike at, indicating the nucleotides that were present. Then this is the result from the mega, okay, which shows us the area of alignment. You can see area of alignment of the C's, okay, showing alignment uh, in the DNA as we compare the two DNA of both the Shodo and the Ataka ethnic group. So you can see that there is alignment in this ethnic group. So these are the haplogroups that were able to be noted. Okay, the haplogroups noted in the Shodo and the Ataka ethnic group. So these haplogroup are similar to also the haplogroup that are noted mostly in the African settings. Okay, so these haplogroups noted are similar to both the Esholu and the affected ethnic group, both for the hyper variable 1 and 2 segments. So we can see the hyper variable 1, the polymorphisms, okay, obtained. Okay, it also show similarity for both the Esholu and the affected ethnic group. Okay, with all the restricting enzymes, okay, acting on similar sites of both the ethnic groups. This is a table showing the diversity, okay, diversity. And we can see that there is no much diversity between the Esholu and the Ataka ethnic groups, as they both show the major L subhaplogroups, which is what is noted among the two ethnic groups. Okay, other haplogroups was not noted among these two ethnic groups. So for the FST distance showing, okay, is there any significant distance in terms of relationship between the Esholu and the ethnic group? We can see between them there is 0.003 FST distance, which is very insignificant. Okay, very insignificant. Okay, so it actually shows that there is a minimal distance, very minimal distance. So there is a close relationship, okay, between the Asholu and the Etekiel ethnic group. So we try to compare it with other ethnic groups in West Africa, okay, at least Example now for the Osholio and the, let's say, the Goro or other ethnic groups like the Tuareg, East Africa, or Somalia, we have 0.057. But you can see for the Osholio and the Teka ethnic group is 0.003. Okay, so we should close that. There's a close relationship between these two ethnic groups. So these are the major L clads that we are noted in the Asholu and the ethical ethnic groups from the subjects obtained. We can also see the L haplogroup is the one that is mostly noted. So we compare this study with other studies done. In this study, we only focus on the Asholu and the ethical ethnic group. Teamwork et al. Did similar work on the Atia, Paju, and the Guru. Okay, so we're able to compare with that. We see that of all other ethnic groups, even the outside, the Yoruba, Nigeria, that we are done. Okay, the Eshodio did not show similarity with any of these as with the attacker ethnic group. Okay, so that is to show that the more similarity between these two ethnic groups. The analysis of variance, okay, also corroborated these similarities between these two ethnic groups. Neutrality test that shows if there's any deviation from the normal also did not show any divergence between these two ethnic groups. So this is the neighbor joining tree, okay, which shows, you can see from the tree, the Ashodo and Eteka ethnic group are closely related, okay, but they are far apart from the Hausas, Yorubas, Nigeria, or other tribes. Okay, so it shows a very close relationship between the Ashodo and the Eteka ethnic groups. Also, the principal component analysis. So, okay, you can see the Ashodo and Eteka on the same quadrant, which are far apart from the European and the Asian. Okay, so you can see that the Esholo and Etekera are in the same quadrant. So have there been any differences between in genetic structure, between the two? Perhaps we would have seen the Esholo in another quadrant, different from where they are found here. Therefore, this study is able to provide evidence of a close genetic tie between the Sholu and the Ataka ethnic groups. And we're able to see that this shared genetic relationship between these two ethnic groups is reflecting in their language, culture, the similarities in their culture, okay, which shows a very common origin, even though these people say they are different. But this study is able to show that they are very similar, and as well as because the presence of gene flow between these two ethnic groups. Yeah. Thank you for listening to this presentation, and I hope you have a very nice day. Thank you.